Yes, thanks a lot for the invitation. I work at the Max Planck Institute for Biology of Aging, which is across the street from Björn at the CCAT. And the talk today will be about the genetics of human longevity from variant to function, where I will show what the genetics of longevity has brought so far and things that we should do different, I think, in the future to get more to an idea of what is actually in the genetics. So as probably all of you know, this is a representation of the lifespan curves from different populations in the Netherlands. So we have the 1850s, 1900s, and 1950s. And as you can see, the curves really shift to the right. So we have been able to really extend lifespan in the general population, mean lifespan. But of course, we haven't been able to extend health span as much as lifespan. So really the goal is to also now find ways to not only maybe extend lifespan, but really also try to extend health span. And very interestingly, if we look at the extreme end of the spectrum, we actually know that this is possible. If you take this very long-lived individual, so individuals above 90 years or above 100 years, we actually see that those individuals have a lower disease prevalence and really show compression of late life morbidity. So it's kind of a natural case study that we can use to actually identify mechanisms contributing to healthy aging. What we also know is this is partly heritable. So if we look at lifespan, we think the heritability, based on twin studies, is around 15 to 20%. If you look in families, a bit lower, also partly due to assortative mating. If we look at the extreme end of the spectrum again, so longevity, which we consider really a different trait, then we don't know what the exact heritable component is. The problem is we do not have enough individuals to actually have a good estimation. However, what we know is if we look at the 10% longest lived within a bird cohort and take their children, then you really see nicely that it is inherited as a trait, as a genetic trait. So we know it is heritable, but we also know it's complex. So we don't believe that there is one single genetic variant explaining it all. We actually believe that there are probably multiple genetic variants, each having a small effect that in combination could determine why a certain individual lives long. And here we have an example of a long-lived family from the Life Longevity study, where you can actually see that they manage, all four of them, to reach an age above 90 years of age, which shows that this kind of thing is really probably in their genes. So there are different ways in which we can study the genetics of longevity. So we can look at the common genetic variation, and we can also look at rare genetic variation. And I start a little bit with an overview of what we have found for the common genetic variation. So we did this large genetic association study where we compared this long-lived individuals, so in this case, individuals above 90 years. Later on, we went to the 10% longest lived for the bird cohort, so we used different criteria. And each time, the only thing that popped up all the time is ApoE. So ApoE is very strongly associated with longevity. And what we know is if we look at ApoE, we have the ApoE2 allele and the ApoE4 allele. And here's an overview of all the different larger genetic association studies that have been done. Most of them have been European, but there's also one in East Asia, and all of them pick up ApoE. There are also some other genes that we pick up, but they normally are only seen in one of the studies that have been performed. So they are not as consistent as ApoE. And then if we look into ApoE, there are two different genetic variants in ApoE. You have the ApoE4 allele, which is associated with a decreased risk of becoming long-lived. So they have odds ratios below 1. And this is really consistent across all the different populations that we test. And on the other hand, we have the ApoE2 allele, which seems to be protective. So it seems if you have this variant, you have an increased risk of becoming long-lived. And of course, as many of you probably know, this is also ApoE4 is also a risk allele for cardiovascular disease and for Alzheimer's disease, while ApoE2 does the opposite. So this is really a disease-causing allele, and this seems to be maybe a more protective allele, which we could say a longevity allele. So since the genetics of longevity was not so successful, I teamed up with several people working on other genetic phenotypes. So they looked at health span, which is the time before the occurrence of the first chronic disease, and there's also genetic data for that, and parental lifespan. So people looked into the data of the parents of individuals, how long they lived, and did a GWAS on that. And if you compare the three traits, you actually see that they are genetically correlated. So you can actually take all the genetic variants for the trait and actually look at the correlation between the two traits. And when we do this, we see that health span and lifespan are correlated with a correlation about 0.7. For lifespan and longevity, that's a bit bigger. It's around 0.8. But for health span and longevity, it's only about 0.5. Still, this is really quite high. And if we then look at the different kind of diseases, also again looking at genetic correlation, so where is the overlap with with specific diseases, we actually see that all of the 3 traits are, for example, correlated with coronary artery disease, inversely correlated with coronary artery disease and stroke, and also with type 2 diabetes, while years of schooling seems to be protective for all these different kind of traits. There's also very specific things that we see. For example, cancers pop up specifically inversely correlated with health span, while Alzheimer's disease is only genetically inversely correlated with longevity. This again shows that there is a commonality between these 3 traits, but there's also differences between them. And we then decided to try out, okay, what happens if we take all these 3 traits together and do one big analysis combining all of them. And when we did this, we ended up with this list of specific genetic variants over here. One interesting one to mention is FOXO3, because FOXO3 was already described in the literature as being important for longevity, genetic variation in this gene, also in humans. We never picked it up in the larger genetic association studies until now, where we actually also found that genetic variation in this gene is important for this combined, we call health span related trait. What we then did is we looked, okay, what do these genes influence regarding specific expression of genes in the blood? So you have the cis genes, which are genes that are in the direct, on the same chromosome, or you have trans genes, which are genes which could be in completely different chromosomes. And what we can see is that there are some of these genes that influence the gene, there are specific variants which influence the gene in which they are, like FOXO3, and also CDKN2B. But there's also some of these hits that seem to influence genes which are much further away. We then looked which of these genes are actually also influenced by aging. So there's actually quite large studies where people looked at the transcriptomics of age in blood, and there you could actually see that there were several genes that were influenced by aging, for example, FOXO3 again, and several other ones, but what was very interesting is these specific genes. They belong really to the top age-related genes in blood. These are really, 5 of them are in the top 10 age-related blood in genes. And what we can see is that with aging, normally these transcripts go down, but however, if we have this specific genetic variant in this intronic non-coding RNA, we see the opposite effect. So it seems to be that this specific variant upregulates then the expression of these genes. So this means it's kind of a master regulator of age-related gene expression in blood. How this works functionally, we do not know yet. This is all bioinformatically predicted, but this is something that I would also like to study in the future in my lab. But it shows what we can do when we use this kind of approaches. Another thing that was quite interesting when we took these genes that we identified and plugged them also in a pathway-based analysis, there was one really strong hit popping up, namely heme metabolism. So we could really clearly see that the genes influenced by these 10 genetic variants that we identified seem to really influence heme metabolism. Again, we don't know how this functionally works, but this is something that I would say we should really try to identify also in the community to see, okay, what is really the influence of heme metabolism in longevity and aging. Another thing that's often asked by people is, okay, if these individuals do not carry so many variants that you can detect with GWAS, how it is then with disease risk alleles? Are these people kind of having less disease risk alleles? Are they protected against them? And it seems to be more of the latter. Because if we look at the disease risk alleles for different kind of diseases, so the people have looked at Alzheimer's disease, cardiovascular disease, many different traits, the only consistent observation that they have is that there is a slightly lower so-called polygenic score for Alzheimer's disease. So they have slightly less genetic variants associated with Alzheimer's disease. However, for many of the other traits, we don't see this. So which means, and on the other hand, if we look at people, if we look at very specific risk alleles, really pathogenic variants that have been identified in childhood diseases and all these kind of things, also long-lived individuals carry those kind of risk alleles. So it seems they might have a slight reduction in some of this genetic risk, but actually they probably have mechanisms that protect them against age-related diseases. So they can, for example, carry ApoE4. ApoE4 is also still present in some of these long-lived individuals, but they can then have something that protects them against the negative effects. So then some things that I think we should really do when we look at the more genetic studies of longevity, we should try to use different kind of approaches. And one thing is that we should ideally focus more on multi-generational families. So far we have looked at singletons, but ideally I think also based on the heritability, we should focus more on the individuals which actually have a really strong genetic component, and hopefully that will help us to identify more genetic variants. Another thing is that we should ideally try to focus on healthy long-lived individuals, so that not only lived to 100 years of age, but were also still quite healthy at that age. And actually there are quite a lot of those individuals. Another thing is we should really try to include more diverse populations. A problem is that we really focus our genetic studies on European individuals, a little bit in Asia, but there's really a lack of people from other continents. And this is also exemplified if you look at ApoE4 prevalence, for example, in the different populations. So as you can see, this is the more rare it is, the higher the prevalence. It's very prevalent in Africa and Central America. However, we have no clear evidence that those people have more Alzheimer's disease or more cardiovascular disease. Actually, these people often die already much earlier. So while in Europe, where the effect of ApoE4 is very clear, the prevalence is actually not so high. And this also shows that there's probably even a gene-environment interaction even for ApoE4, which is so clearly associated. And we expect that for also many other variants that we identify. So it's very important that we really also measure these kind of things in other populations, because we cannot just generalize things based on the European population when it comes to genetics. Another thing is that we can try to use alternative approaches for analysis. So we can do kind of linkage analysis, going back to long-lived families, where you can really look specifically in these families what is inherited. Or we can look at gene-gene and gene-environment interactions. This is really what I would love to do more. The problem is we don't have the power really to do this. So this is really difficult. But we also, proof of principle, we already did this in a very small population. And I have to say, in the Croatian population, where we had individuals above 85 years of age, and what we did is we took some of the SNPs that were previously associated with lifespan or longevity, and we looked actually at the interactions. And what we saw is that there is indeed interactions between some of these SNPs, and this is highlighted over here. So if you look here in the yellow, those are individuals that carry a combination of two of the variants that are potentially protecting against disease, and they have an increased or decreased risk of dying compared with individuals that just carry one of these alleles or none of these alleles. Like I said, the power is not so big, but it shows that the approach can work. So in the future, I would also like to go more into this direction. Another thing that I have been doing when moving to the Max Planck Institute for Biology of Aging was actually starting to focus more on variants that are a bit more rare in the population. So we have seen the large GWASs. They come up with ApoE, but we felt there is more to gain, right? So it's maybe more in the rare genetic variants where we have to look. However, there's a problem when you start looking at rare genetic variants, because you cannot prove their causality in humans. So when you do genetic analysis, you have, for example, 2 individuals that carry these alleles versus none of the controls. There's no statistical test that can accurately address this. So we really need to do more functional studies. And I use kind of reverse translation, the reverse translation approach, where I come from the human side and go back into the model organisms. So we really do functional studies in cellular models and also in model organisms, and in this case, mostly in mice. So the idea is we look first into pathways that have already been associated with lifespan in model organisms, and then we look into the genes in those pathways in humans, and look if those long-lived individuals from these families, but also singletons, have specific genetic variations in those genes, and then see if they are functionally influencing the pathways in which they reside. And this approach will likely work because it has already been successfully applied by others in the field. So Yusin Su's group, for example, she has already did a similar approach for the IGF-1 receptor. and variants in SIRT6. And there have also been approaches where people looked into the effects of genetic variants in FOXO3. And this is an example of the data for the IGF-1 receptor, where at that time they brought in this specific variant and they looked at the phosphorylation of IGF-1 receptor, and they could actually see that these variants that they brought in lowered the phosphorylation of IGF-1 receptor and also downstream mTORC1 signaling, mTORC2 signaling, sorry. So we think that this approach can work, and this is also why we went ahead with it. And as the first step, we really do the in vitro characterization where we use the cellular model. But of course, the question is first is how do we identify exactly the variants that we are working with? So we have two different study populations. We have the longevity study, which is a family-based study. So we know also multi-generational that these families are long-lived. And we have actually another study from Germany, which is the German longevity study, which contains this so-called long-lived singletons. So these are individuals without a known family history for longevity, but these individuals lived a bit longer. So most of them reached an age above 100 years. So how we then selected the variants that we focus on, like I said, a gene or pathway coming from model organisms. We look then for protein-altering variants, so that's in the coding region of the gene, and they have a so-called CADD score of above 20, which means that it's very likely from a bioinformatic point of view that they are associated with the functioning of the protein. And what we also did, we focused on variants that are absent or extremely rare in the general population to increase the chance that they can actually contribute to the longevity phenotype, which is very rare in the population. And what we then did is that we generated and characterized transgenic cell lines in mice, and we generated them via CRISPR-Cas9. The focus was, we decided to really focus on one specific pathway, which is not surprising. So we summarized actually the literature recently and looked, okay, what is the genetic evidence coming from model organisms and humans of pathways that could be involved in lifespan regulation? And for example, the DNA damage and response pathway and repair pathways there, but really the clearest hit is, of course, in the insulin IGF-1 signaling pathway. So this was also the pathway that we decided to focus on first, also more as a proof of principle that our approach can work. And I don't think I have to explain many of you how this pathway works, but what is important to know, of course, is that you have the mTORC1 complex and the mTORC2 complex, and most of the beneficial effects of lifespan extending interventions in model organisms seem to run to mTORC1, and some of them also partly to this MAPK-ERK branch of the pathway. And what people have normally seen is when they look at the phosphorylation of S6K, they see a downregulation, and also a downregulation of phosphorylation of ERK. So these are kind of the two things that are generally seen when people look at lifespan extending interventions in model organisms. And interestingly, this is also what we are seeing. So first, shortly, how we generated the cell. So we use a haploid mouse embryonic stem cell system. So it's a haploid system, which means we can easily target it with CRISPR because we just have to edit one allele. Another advantage of this system is that it provides direct translation from humans to mice. So the variants that we have been using are conserved from humans to mice, which means that when we see interesting effects, we can actually make mice containing the exact same mutations. And when we then look at the mTORC1 signaling and also at mTORC2 signaling, when we look at S6K phosphorylation, we see it's downregulated in all of our cell lines. However, you should be aware that this effect is actually quite mild. If you take rapamycin, for example, you would see a complete shutdown. And this is also not surprisingly. We didn't expect that these people would carry variants that would shut down the pathway completely. We actually expected that the mutations would have more mild effects. And when we look at AKT, we actually see a distinction between the cell lines. So some of the cell lines do not show any difference in the phosphorylation of AKT, and some of them seem to show an upregulation. And this is probably correlated to the growth of the cell lines, which I come back to in a minute. We also looked at the phosphorylation of ERK, which is, like I said, part of this MAPK-ERK branch of the pathway. And also there it has been shown that a downregulation of this pathway is considered beneficial for longevity. And we see actually that all of our mutants also downregulate this part of the pathway. So the effects that we see here might hint that these variants be beneficial for lifespan. We then did untargeted transcriptomics, proteomics and transcriptomics. And what we then noticed, and it was a very clear effect when we just did a really simple principal component analysis, look what explains most of the variation in the data, we see a very clear clustering. So we see here a cluster of 3 of our cell lines, and here a cluster of all the remaining ones. And they're all different from the wild type, which means that this is really a big effect that we are seeing. And we're seeing it both in the transcriptomics and matching exactly the same clustering in the proteomics. And also when we look at the overlap, because there's often a lot of discussion, are proteomics and transcriptomics overlapping well? In our case, it does. So actually, when we see specific transcripts being downregulated or upregulated, we see it in both in the transcriptomics as well in the proteomics. So there's also a really good correlation between the two of them. So we are currently looking, okay, what are these groups representing? This is still work in progress, but we're trying to figure out, is there something in this group that can explain why they are like this? And one thing that I can already tell you, what was a very clear observation, when we looked at the growth of our cells, we actually saw that this matches nicely with the grouping. So you have, for example, this is of the cells that are in group 2, they seem to grow a bit faster. And this matches again well with the data that we have seen for the phosphorylation of AKT. If we look at the other 3 variants, they come from the other group, so the smaller group, they seem to grow a bit slower. So there seems also to be a correlation between the grouping and more phenotypic effects. We also see effects on stress resistance. I don't have the time to show that today, but we actually really see this clustering in the grouping not only at the omic-based level, but also at the phenotypic level in the cells. But then I want to switch to the second part, which is namely that in vivo characterization in mice. So we actually quite quickly went ahead with some of these variants and decided to bring them also in mice to see what the effects are there. Again, we use CRISPR-Cas9 gene editing, and be aware, these mice contain just one mutation, right? So the mutation that we identified in the humans, this is the only mutation they have in their whole genome. We look at the lifespan of these mice, and we do detailed phenotyping. So we measure different kind of phenotypes, which are often measured in the field, like grip strength, frailty score, insulin signaling, glucose signaling, body composition. So kind of the standard phenotyping pipeline. And we do that during 3 times in their life, 4 months, 15 months, and around 22 months. And we also collect tissues from the mice. So we have actually different cohorts where at the same time points, we also collect tissues. And we do that in a normal state, and also we fast the mice and then give them food so that we actually can collect tissues in a more stimulated state. And one thing that we noticed when we were breeding these mice, and this was a very clear effect, is that the litters of the mice are bigger for at least 2 of our variants. So when you look at the litter size of the different mouse strains, the 2 of them really have 1 or 2 mice more per litter when they give birth. So which is a really striking effect. We don't know where it's coming from. This is something that we are looking into at the moment. So we took the testes and ovaries of these mice to see if we can explain it. But it was a very striking effect, which we didn't expect. When we then look at body composition as one of the things that we are doing, we see that for one of the lines, we actually see that the homozygote mice are a bit smaller at young age. And interestingly, this seems to disappear when these mice become older. So it seems to be a developmental effect that these mice are having. And it's sex-specific. We only see it in the female mice, not in the male mice. And this is really important to realize that many of the effects that we are seeing are sex-specific, which is also the reason that we took along both sexes, because we expected that this could actually happen. And this is also exemplified by one of the other lines, where we actually see an effect in the males. So we also, again, look at body weight. We don't have the old time point yet. We are currently collecting. But in the middle age time point, there we start seeing a difference in the weight in the male mice. We have to see if that holds in the old time point, but this would be more of a longer-term effect, so not a developmental effect, but something that appears only later in life. So these mice seem to maybe age differently. We also look, of course, at a lot of phenotypic measurements, and I want to give you one example over here, which is for motor coordination, which you can measure with a so-called rotor rod. So you put these mice on these machines. The machines start turning, and you measure how long they can stay on the machine before they drop off. And they have a learning phase, so it takes them a couple of days to learn how to use the machine. And during that time, you measure how well they are doing. And when we look in the young age, again, female-specific for this mouse line, we see that they are doing better on the rotor rod. The effect is still there, not significant when we correct for multiple testing in the middle age, but it seems to still be there. It disappears in the old age, and part of that is probably because the mice that are really performing badly, they die. So actually the wild-type mice become better because the bad-performing ones are dying. And how that looks, if you look a bit more into this specific time point, you see that here are the different days on which the mice are on the rotor rod, and here's their so-called latency to fall, and you really clearly see that our mutant lines are doing better. But of course, the most interesting thing is the lifespan data. And this is the data from 2 of the lines for which we already have sufficient lifespan data, both in females in this case. We see a potential lifespan extending effect. Especially in this case, it seems to be, at the moment, significant. We have to see how that turns out in the end. But we don't see anything in the male mice. We just see this in the female mice. Be aware, I say it again, this is one mutation that these mice have, which makes them different from wild-type mice, and we could really nicely already see effects over there. When we zoom in a bit of what could be the reason for that, one thing that we observed is that the wild-type mice, in this specific Black 6 mouse line that we are using, die of, we have to kill them because of UD, ulcerative dermatitis. So they get this infection of the skin, and when they get this, they need to be sacrificed. And we see actually that the onset of this UD is much later in our mutant mice. So this could be the reason that they live longer at the moment, especially at the younger ages. So the conclusions are that the genetic component of human longevity is complex, and it's likely determined by many different variants, probably rare variants, that each have a small effect. We have successfully generated transgenic mouse lines and also mice carrying protein-altering variants that we identified in long-lived individuals. These transgenic cell lines show changes in the expression of the genes. I didn't show that today, but really also in the genes itself, which harbor them, but also in insulin, IGF-1 signaling, and mTOR signaling. They show overlapping transcriptomic and proteomic differences, but also distinct between the 2 groups. And some of these transgenic mice show some promising beneficial phenotypic effects, such as motor coordination, and we are still in the process of analyzing them further. So there are many people that work with me on the large genetic studies of longevity. These are just the analysts that I work with. And of course, very importantly, my group at the Max Planck Institute for Biology of Aging, the group of Eline Slagboom and Almut Nebel, who are sharing the genetic data of their individuals with me so that I could analyze it. Of course, the people from the core facilities in our institute, which are very important to run all the mouse studies, for example. And last but not least, the funding agencies. Thank you for your attention.
Thank you very much, Joris. Questions? Bjorn.
Joris, I wonder, do you see actually, with regards to the ulcerative dermatitis as an outcome, do you see any differences in inflammation? Does it ameliorate age-dependent inflammation, the mutation?
So we haven't checked that yet. What we have just done, we sent the samples for Olink for 2 of the mice lines, where we actually look at inflammatory markers. So they have a new cytokine panel specifically for mice. And we have measured this now in the blood of the mice. And we have to see what we see there. We also still have the tissue, so we can look at inflammation. But for this line specifically, we haven't done that yet. No, it's something that we would like to do. Yeah.
Going back to your GWAS analysis with the representation of the heme metabolism, do you have any thoughts on specific hits there, or like what mechanisms might be involved?
So the only thing what we know there is that it's driven by the hits that are coming from genetic mutation near the LDL receptor. So this LDL receptor mutation seems to influence many genes, and these genes, when you analyze them together, seem to influence heme metabolism. How it exactly works functionally, we don't know. This is just an enrichment analysis. We haven't looked specifically in how the pathway is regulated. It would be something that I think we should look into more, also maybe in human cohort data, but we haven't addressed that at the moment yet, no.
Any more? All right, thank you so much.